Okay, hey guys. So the goal today is to do a makeup lecture for those folks that weren't able to attend last Thursday when we talked about carbohydrates and lipids. So this is my attempt to get you guys caught up. So I'm here in my office. I've been on my feet for 8 hours teaching, so I'm being pretty casual right now. I'm on my Stegosaurus beanbag chair in my office at NAU. All right, let's get this party started. Share my screen. Oh, I gotta push this button over here and pull this up. Okay. So, as you guys are prepping for your exam, you guys want to be able to answer these questions fully. So, what is the role of carbohydrates in and outside of the cells? Um, how do we classify carbohydrates, and what are some common monosaccharides, some disaccharides, and some polysaccharides? First of all, what the heck do all those words mean? I will tell you in a minute. Okay, so here we've got some examples of some common carbohydrates you guys might be familiar with. Simple sugars, we've got some table sugar, we've got some fruit that's usually high in sucrose and glucose and fructose. We've got some complex carbohydrates. So you guys are probably familiar with carbs as dietary carbs, being bread and pasta and rice and all those delicious things that I love. But maybe you don't realize that the components of wood are actually also complex carbohydrates. So wood is made of cellulose and lignin, both of which are carbohydrates. So carbohydrates basically, so these are just carbon. If you take the word apart, carbohydrate, it means carbon and the junk that's in water, right? So if we look at this, this chemical formula here, we can easily see the water part, right? H2O. And then we almost always, but not always, find, oopsies, find the same number of carbons and oxygens, and then twice as many hydrogens. And so that's, we can use this chemical formula to say, okay, if I told you this sugar molecule has 6 carbons, you could say, okay, that means that there's 12 hydrogens and 6 oxygens as well. Okay, so you just distribute this N value. Oops, come on, mouse. You just distribute this N value to everything inside those parentheses. Okay, so let's talk about monosaccharides. These are the simplest sugars. Mono means one, saccharide means sweet, so these are just one sugar molecule. These are our monomers. When we start talking about linking these together to make complex carbohydrates, our monosaccharides are the monomers for carbohydrates. Typically, some kind of general rules, we usually see 3 to 6 carbon atoms in monosaccharides. We usually put the ose as the ending of the name, so glucose, fructose, cellulose. That ose kind of like bings your brain into thinking about carbohydrates. And then there's some other naming conventions that tell us where the carboxyl group is and how many carbons there are. These are details that is just for your information, but you don't get too bogged down in the details for our exam on Thursday. So what we'll see, we'll look at these two. These are structural isomers. So they have the same chemical structure. They have 3 carbons, 6 hydrogens, and 3 oxygens. Okay, so they're isomers because they are put together differently. The arrangement of the atoms are different. So this is showing you basic differences between this really broad category of aldoses and ketoses. So the way that I remember an aldose is that on the terminal end here, on this terminal carbon, that's where we have our oxygen bonded, double bonded to the carbon atom. And so I remember this by saying Uncle Al has terminal cancer, and then I can remember aldose, that double bonded oxygen goes on the end. A ketose, on the other hand, has that double bonded oxygen, one carbon in from the very end. And in this case, because it only has 3 carbons, it's double bonded to the middle. Okay? So just kind of know maybe the big difference between an aldose and a ketose. Ketoses behave differently in our bodies than aldoses because of that location of that double bonded oxygen. So this is just showing you that 3 sugar molecules that have 3 carbons, we give them the general name of triose, right? This is just a broad category. Every 3 carboned sugar has the name of triose, but we can get more specific to say that this is a glyceraldehyde. You don't need to ever remember that word as far as I'm concerned. But triose tells you 3 carbons. Pentose, that's 5, like think pentagon, right? That's 5. And it's ose, it means it's a sugar. Hexose, that's a 6 carboned sugar. The one that we see the most often is glucose, because that's the one that we care about the most, because it is really it's really important for energy production and movement across. It's a precursor for a lot of our biomolecules and our metabolic pathways. Okay. So let's talk about isomers for a minute. So the first thing that we need to do is remember what the heck isomer means. So isomers are molecules that have the same chemical formula. So in this case, it is 6 carbons, 12 hydrogens, and 6 oxygens. So all of these molecules, glucose, galactose, and fructose, all share that same chemical structure. Let's see if you guys can see this. No, you cannot. So I won't even try. But what it means, the reason that they're different, even though they have the same chemical structure, is that these atoms are put together differently. And so if we compare glucose and fructose, you can easily see that where that double bonded oxygen is in a different spot in glucose and fructose. Those are our structural isomers. If we look at glucose and galactose, however, we will see, come on mouse, that they're very similar in structure. And it kind of takes us a minute to like figure out where the difference is. But if we look at carbon 1, 2, 3, 4, we look here, we see that this hydroxyl group is on different sides, right? So that's the difference there. Just remember, this is just an example of isomers. You don't have to memorize the chemical structure for these things. So everything that we've looked at so far has been in this linear structural shape, but what we will also see a lot of the time, especially with these monosaccharides, is that they're usually have this ring form. And so again, don't get too caught up on the chemical structures, but just know that, like, be able to recognize that if I show you this little like circle thing with a bunch of carbons and hydrogens and oxygens sticking off of it, you're going to be able to say, oh, I know that's a carbohydrate. Okay. And so these sugar molecules switch between its two ringed forms and its linear form in your body. Functionally, it doesn't really mean a lot, but you'll see it drawn both ways. And so the reason I'm telling you is this so that you don't think that this molecule and this molecule are different. It's just a different way to draw it. Okay, so there was a lot of folks that were getting hung up and confused on this. So this is one of the questions that you guys had. So it was about the numbering conventions. So what we're going to do in order to know what carbon we're talking about, and like you guys saw today, This is important when we talk about DNA replication. So what the first thing that we got to do when we see this ring structure is that we got to find the oxygen. And then if we just go clockwise, we can start counting the number of carbons. So remember that even though there's not a C here, we know that that little bendy place in that chemical structure, that there's a carbon hiding there. So we're going to call this carbon 1, and then carbon 2, and carbon 3, carbon 4. And then this is where that double bond comes into play, is because now this is sticking up outside. It's kind of like a little flag sticking up off the ring structure. This whole group right here. Okay, and so that's where our 5th carbon is. So just kind of be able to be familiar with this numbering convention, and because that allows us to talk more easily about which carbon is doing what when we start talking about DNA replication and other things like that. Okay, glucose, this is the rock star of the carbohydrate world. This guy is soluble in water. It's easy for us to break down. The energy that is released in the breakdown of glucose is transferred to a molecule called ATP that we'll talk a lot more about. But this is, so I talk a lot about biology and ecology in terms of economics, because really what we're trading in is energy. And so ATP is the currency of life. This is the money. ATP is where the money is, and glucose is how we earn the money. So glucose is a really important molecule. Okay, so that concludes the blah blah about monosaccharides, and now we can move into disaccharides. Disaccharides are 2 sugar molecules hooked together, right? 2 monomers hooked together to form a dimer, because it just has 2, or you could say call it a polymer. Either way would be correct. So the disaccharide that we're most familiar with is probably sucrose. This is just plain old table sugar. And so this is a glucose molecule bonded to a fructose molecule. So we had a dehydration reaction occur. A water molecule was pushed out, and then this bond right here was formed during that dehydration reaction. Okay, we can easily break these apart using hydrolysis reaction by adding a water molecule and releasing that stored energy that's stored in that bond. Again, don't get too worried and hung up about the chemical structures. I'm not going to say, give you a chemical structure and say, okay, tell me what sugar molecule this is. That's not what this class is for. You might take other classes for like that in the future, but that's not for my class. Okay. But let's talk a little bit more about that bond. The glycosidic bond for 2 monomers of carbohydrates, we give them the name of monosaccharide. When they get together, they form a glycosidic bond. This is a special kind of covalent bond. It's formed by the dehydration reaction between 2 carbohydrates. And so we can see here's our OH group, and here is the hydrogen that's going to be donated. And then this oxygen is going to be the one that is forming the glycosidic bond. And that other water molecule is pushed out during that dehydration reaction. Okay, and it also requires, dehydration reactions require energy to be input in order to make that bond. Some other things that we might see very often, and again, don't get hung up on the circles and the letters and the little things, and but you probably heard of maltose before. Maltose is malt sugar. It's 2 glucose molecules bonded together in a glycidic bond. We're seeing that glycidic bond right here, boop, around that oxygen atom. And then our lactose is a glucose molecule with a galactose molecule in forming a beta bond. And I'll tell you about those in a minute. So you've heard lactose before. If you're one of those lucky people that gets to be lactose intolerant, is that you don't have the enzymes to break that particular bond. And so that's what causes your tummy to be upset, because your body's trying to break this down, but it can't. And some other products are being produced instead of a glucose and a galactose molecule. Okay, so now we can talk about polysaccharides. Poly means many. We're still talking about sweet things. So saccharide meaning sweet. Our glucose monomers can hook up together in these really long, complicated things that store energy. In plants, we call that starch. And in animals, we call that glycogen. Okay. And it's the difference between starch and glycogen is just how all those bonds are formed in arrangement with each other. So what I need you to remember is that starch equals storage for plants, glycogen equals storage for animals. Okay, and we can tell that this is a polysaccharide because here's that circle. It's kind of this weird thing. It kind of looks like this is the chair formation. I don't know if it really looks like a chair. It doesn't really to me, but I don't know. Maybe I'm not using my imagination correctly. But that's that circle. That's the glucose ring of carbons. And so there's a bunch of them hooked together. And so because there's a bunch of them, we call it a polysaccharide. Starch and glycogen, like I mentioned before, these are used for storing energy. It's an easy way to store a bunch of these glucose molecules. They're easy to break down, and then we can release energy from that glycosidic bond and use that energy for whatever we need to use it for. And then we can also break down those glucose molecules in order to produce the money, the ATP. So that's one of the jobs of polysaccharides is for storage. And another job that polysaccharides does outside of the cell is that it's used for structure. And so cellulose is a bunch of glucose molecules hooked together in a beta bond. And that's one of the reasons that wood is hard, because of this structural polysaccharide carbohydrate. Bacteria have a certain kind of polysaccharide that they use in their cell walls for protection and for structure. Fungi and insects use chitin. This is a little bit sugar, a little bit protein mashup. And that's used for protection in their cell walls and in their exoskeletons. And then us animals have this really big, long, scary name of glycosaminoglycans. I don't know. But that's what creates our connective tissues that allows us to move our muscles and stuff. Okay, so polysaccharides, storage, energy storage, and structure. That's the takeaway for that. Okay, so what I'm going to do is I'm going to stop this video now, and I'm going to create a whole nother video for the lipids. So there'll be a little bit more bite-sized, manageable pieces. So I will go and do that now.